Specimen 23 أول سؤال. Catalase is an enzyme found in the plant and the animal cells. It catalyzes the breakdown of hydrogen peroxide to form water and oxygen. The oxygen produced forms a foam. You can measure the height of foam to determine the catalase activity. يبقى in this experiment, how are we going to measure the results? We are going to measure the height of foam. A student investigated the catalase activity in cooked and uncooked potato tissue. This is what's going to be different. This is the changed variable. Cooked and uncooked potato. This is the changed variable. Okay, what about the factor we measure اللي هو ال dependent variable? الريزلتس بقى. The results that I'm going to measure is the height of foam. يبقى this is the results to be measured. طيب هنعمل ايه؟ The student cuts the two potato cylinders so that they are identical in shape and size. Okay. And then he puts 5 cubic centimeter of 3% hydrogen peroxide into a test tube labeled cooked potato. Put 5 cubic centimeter of 3 hydrogen peroxide into a test tube labeled uncooked potato. طيب I put the same volume of hydrogen peroxide and the same concentration of hydrogen peroxide in both test tubes. Since they are the same, يبقى these are the controlled أو fixed variables that are constant. أهم الفوليوم والconcentration in both test tubes the same. الاتنين زي بعض. So they are the controlled or the fixed variables. Put one of the potato cylinders into a beaker of hot water for 5 minutes. You have one cooked potato. طب انت هتعمل cooking ازاي? How are you going to do the cooking? You will put one of the cylinders in hot water for 5 minutes. Remove the potato cylinder from hot water and put it in a test tube labeled cooked potato. Put the uncooked potato in a test tube labeled uncooked. طيب ايه المشكلة؟ What is the problem here? احنا when doing an experiment everything should be the same. يعني since you soaked the first potato in water, the second potato should also be soaked in water. بس ممكن بقى احطها in cold water not hot water. Okay? طيب left the potato cylinders in 3% hydrogen peroxide solution for 3 minutes and then measured the height of foam produced in each test tube. Okay, you left both potatoes in the hydrogen peroxide for 3 minutes. يبقى this time التايم ده برضو controlled variable. Okay, this is the height of foam. It shows the test tubes after 3 minutes. Potato cylinders اهي, and this is the height of foam. Okay, and he puts a ruler next to it to measure it. عايز ايه بقى? What am I going to do? Prepare a table to record the results. Measure the height of foam in each test tube. Record these measurements in your table. The table زي ما اتفقنا كل مرة it's the same thing. The changed variable is on the left والresults on the right. طب ايه ال changed variable؟ ال treatment of potato أو ال cooking of potato. مرة كانت cooked ومرة كانت uncooked. ايه الريزلتس اللي انا بقيسها؟ ال height of foam. You're only measuring the height of foam. يعني you should start from here to here. هو ده الفوم. This is the foam height. Measure it using the ruler next to it. Not the whole height. ده hydrogen peroxide. أنا مش عايزة أقيس ال hydrogen peroxide. I only want to measure the height of foam. So how will the table look like? It will look like this. Potato treatment, uncooked وcooked. On the other side عندي ال height of foam / millimeters. We agreed إن أي حاجة في paper 6 it's better to measure in millimeter. So الرولر دي بالسنتيمتر أهي. How to convert from centimeter to millimeter? I will multiply by 10. Okay, after measuring, you will put the numbers here in millimeter. Do not put the unit in the table again. The units are only in the headlines. Okay, calculate the difference in the height of foam produced by the cooked and the uncooked potato. Take care, هنا he just needs the difference. He did not say the percentage change. I'm not multiplying by 100. أنا بس عايزة ال difference. يبقى هعمل إيه؟ الرقم اللي هنا minus الرقم اللي هنا. I will subtract these two numbers only. طيب, state one conclusion for the results. حتى even if I don't have the numbers. إيه conclusion? مين اللي طلع foam أكتر؟ uncooked potato produced more foam. This means إن catalase enzyme was working faster. Yes, it was working faster. يبقى what's the conclusion؟ هقول له إيه؟ Enzyme is more active in the uncooked potato because it produced more foam. Okay. What is the independent variable? بصوا بقى هنا جاب لي كلمة independent variable and he did not say it changed. So you have to know إن independent ده هو ال changed variable. The independent is the changed variable. إيه اللي أنا غيرته في الإكسبيرمنت دي؟ The potato treatment. Why is it important that the two potato cylinders were identical in shape and size? ليه لازم يبقوا identical? Because if they are different shape and size, they will have different surface area. يبقى هما they should be identical, so they have the same surface area because I only changed one thing, which is the potato treatment. I cannot change two variables at the same time. Okay? Okay. What were the two variables that were kept constant زي ما اتفقنا? What were the controlled variables or the constant variables? We said that the volume was the same, the concentration was the same. I can also say the time in the hydrogen peroxide solution was the same. Identify one possible source of error. ايه possible source of error؟ ان قلنا حاجتين. cooked potato was not dried before adding to the hydrogen peroxide. This means that it had water on the outside, which might affect the concentration of the hydrogen peroxide. يبقى أنا لازم أنشفها كويس من بره so that there is no water on the outer surface. أو ممكن أقول إن surface of the foam is uneven. When you're measuring the foam, you will not be able يعني should I measure from here ولا from here يعني it's a bit difficult. That's why there was a range. There was a range in the marking scheme. So when you measure, it's not that accurate. This is another source of error that I can write. A student stated that a catalase activity is the same in all species of plants. Plan an investigation to test this statement. Catalase activity is the same in all species. I want to make sure if this statement is right or wrong. طيب هعمل ايه؟ You will get different species of plant and then you will do the catalase test زي اللي فوق like the one we did before and you will compare. طيب we will write the same steps of design an experiment. أول step is the replica. يبقى number 1 replica. How many plants will you get? قلنا at least 5 at least 3. كل ما كان أكتر كل ما كان أحسن. يبقى use 5 plants. Okay, use 5 plants. Okay, number 1 is the replica. How many plants are we going to use? Use 5 plants. Number 2, changed variable. What am I going to change? Each of them of a different species, because I want to check لو الكتاليز activity في كل species of plants are the same ولا لأ. So ايه اللي انا هغيره؟ I will get plants of different species. All the other variables should be the same. أي حاجة, anything else that affects the enzyme activity should be the same. Same volume of hydrogen peroxide, same concentration of hydrogen peroxide, same temperature, same pH. Okay, يبقى number 3 controlled أو fixed variables. Number 4, what results are you measuring and how are you going to measure؟ يعني مش بس تقولوا لي ايه اللي انا هقيسه، لا هقيسه ازاي هقيس الحاجة دي. طيب in this experiment I can write measure the height of foam using a ruler like this way، وممكن اقول له and this is a better way of measurement measure the volume of oxygen gas produced using a gas syringe. مثلا in 3 minutes measure the volume of oxygen gas produced in 3 minutes. Number 5 write an improvement. ايه الامبروفمنت اللي انا ممكن اعمله في الاكسبيرمنت to get better results؟ Since you will have same temperature, you have to maintain the temperature using thermostatically controlled water bath so that the temperature remains as it is. أنا مثلا عايزاها 35 degrees, it will remain at 35 degrees, it will not cool down. Safety precaution, wear goggles, and then repeat. آخر حاجة repeat at least twice and calculate the average. يبقى ده design an experiment. Okay. The last question هنا, it was very easy, just food tests. Describe the test to identify starch and reducing sugar and give the positive results. الستارش التست بتاعه هيكون iodine. The color changes to blue black. reducing sugar we add Benedict and then لازم heat in water bath. If there is sugar it will change to orange red. Okay. Question number 2. A woodlouse is a small animal. The rate of respiration of a woodlouse can be measured using a simple respirometer. Chapter 11. As the woodlouse respires, the drop of the colored liquid moves along the capillary tube. There is a ruler next to the capillary tube عشان أقيس distance، أشوف هي اتحركت قد إيه. This is the start and this is after 30 minutes. الدروب بقت هنا. طيب أول حاجة بيقول لي record the position of the drop at the start and after 30 minutes. ماشي. هي at the start تقريبا it was 4.5 centimeters. 4.5 centimeter يعني كام مليمتر؟ 45. I will multiply by 10. هنا تقريبا كانت 0.5 centimeter. So I'll also multiply by 10. It will be 5 millimeters. طيب هنا كان في range. In the marking scheme there was a range. If you write 0. يعني the range was from 0.3 to 0.6 I think 356 كده، كان في كده range في النص 0.6 0.3 0.4 sorry 0.4 0.5 0.6 من 4 5 6 millimeters. This was the range. طيب calculate the distance moved by the drop in 30 minutes. اتحركت قد ايه في 30 minutes؟ You will just subtract. هي كانت عند 45 بقت عند 5، يبقى 45 minus 5 it will be 40 millimeters. طب calculate the rate of movement per minute. rate of movement per minute. الرول بتاعت الريت is something over time. أنا عايزة rate of movement of the drop. يبقى I will see the drop moved for what distance? It moved for 40 millimeters في كام minute? In 30 minutes. 40 divided 30, so the result will be, it will give me a number. I should round it to 1 decimal place. It will be 1.3 millimeter per minute. Okay. The rate of movement of the drop of colored liquid along the respirometer can be used to estimate the rate of respiration. The student used the respirometer to investigate the rate of respiration in 4 animal species. 4 species مختلفين, and we're investigating the respiration بتاعهم. أول حاجة قال لي calculate the missing mean. المين هي الaverage. فأنا هعمل إيه؟ I will add all these together divided by 3. It will give me 1.5. بعد كده قال لي plot a bar chart to show the mean movement of the colored liquid for the 4 animal species. عايزة bar chart. البار chart ده for the mean only. طيب, what will I do? First of all, put the labels. The left side on the x-axis, the results will be on the y-axis. يبقى هحط ايه؟ animal species on the x-axis. On the y-axis عندي mean rate of movement / millimeter per minute. محدش ينسى يحط الunit. You have to put the unit. And then you will draw one bar. طيب, مبدئيا you have to put a scale. أنا عندي الأرقام لحد 2.5. So I put 0, 5, 1, 1.5, 2, 2.5. Don't forget to put the dashes to determine where exactly you put the number. And then draw the bars. أهو bar A كان عنده 1.5, هنا 0.9, هنا 2.5, 1.9. Each bar has its letter below it. The bars should be equal in width, and the spaces between them should also be equal. طيب, بعد كده بقى بيقول لي ايه letter of the animal with the highest rate of respiration. طبعا it is C. Suggest a suitable control for this investigation. Control for the investigation أو control for the experiment. يبقى أنا هعمل إيه؟ I will do the same experiment again without the wood lice. أو بدل الwood lice أحط glass beads. It's a non-living thing, مش هتعمل respiration. طب أنا كده هستفيد إيه؟ Why do we do this? قلنا 2 reasons. To make sure, عشان لو سألني, لو جه السؤال بعد كده, why did I do the control experiment? To make sure that the drop moved because of respiration. Make sure that the drop moved because of respiration of wood lice. دي حاجة. الحاجة التانية, and to compare results. يبقى these are the 2 reasons why we do a control experiment. Okay, the student decided it would be better to calculate the rate of respiration per gram so that the values could be compared. يبقى أنا أصل ممكن ال species the 4 species might have different mass, so we should not compare them كده together. طب عشان أقدر compare them قال لي إيه؟ I will calculate the rate of respiration per gram. How can you find out the rate of respiration per gram? Very easy. You will just measure the organisms الأول and then divide the rate of respiration by the mass. يبقى measure the mass of the animal وبعد كده divide ال rate of respiration by the mass. Okay, the last question, ال drawing, it shows a photograph of a wood louse. He wants you طبعا to draw it. طيب how will I draw it؟ أولا أنا عندي projection هنا، two projections هنا و two projections هنا، يبقى لازم أخد بالي منهم. بعد كده ال segments 1 2 3 4 5 6 7 8 9 10. 8 9 10 دول صغيرين. In the marking scheme he said that you should at least draw 8 segments. Any drawing عندها segments كتير, there is usually a range in the marking scheme. طب how am I going to draw it؟ عشان أسهل على نفسي you can do an oval shape كده. Okay, and then start dividing the segments. يبقى لما أرسم الخطين دول draw it lightly، خلي القلم الرصاص خفيف عشان تعرف تمسحها بعد كده. وأقسم بقى الايه السيجمنتس، ادي 1 and then 2 3 okay 4 5 هتنزل بقى على أصغر 6 7 8 9 10 and then امسح بقى الخط اللي من بره ده. اوكي، وبعدين نرسم بقى ايه البروجكشنز بتاعتنا زي ايه، زي الصورة في واحدة. فطبعا انا رسمتها دلوقتي بسرعة، انت يعني المفروض ترسمها بالراحة draw it slowly عشان ايه يجيلي الشكل. ف it should be something like this. two projections in the front and at the bottom and the segments مرسومة مظبوط. طب انا اظبط الشكل ده ازاي؟ how to adjust the shape. Draw an outline with the pencil lightly, draw in it the segments and then erase the outline. طيب the magnification of the wood lice is times 9. Measure the length of the line PQ in millimeters. So you are going to measure the length with the ruler. The ruler will give me centimeter multiplied by 10 to get the هيكون كام؟ 4.8 centimeter. Okay, so it might be مثلا 48 millimeter. Calculate the actual width. قلنا التريانجل اهو magnification equals image over actual. لو انا عايزه الاكتوال then I will do image over magnification. الimage is 48, magnification is 9. Put your answer to the nearest 3 significant figures and this is it.